Good evening, ladies and gentlemen. The next lecture is a new addition to our academic program. It is the SLCE Medal Lecture. This is in memory of Professor Carlo Fonseca, who was the first president of the Endocrine Society of Sri Lanka. So the recipient of this medal, of this inaugural lecture, is Dr. Noel Somasundaram, a clinical endocrinologist. May I now invite Dr. Noel Somasundaram to accept the medal and deliver the lecture.
Thank you, Mr. President. Ladies and gentlemen, we are here today to honor the first president of the Sri Lanka College of Endocrine Society of Sri Lanka, which later transformed into the Sri Lanka College of Endocrinologists. Once my slides come up, you could see the images of Professor Carlo Fonseca. He was born in 1933 in the city of Negombo and had his primary education in Mary Stella College, subsequently in St. Joseph's, Colombo. And then he entered the then University of Ceylon. And he passed out in the early 1960s with flying colors. Can I have my slides up, please? He passed out with flying colors from the University of Colombo with many medals and obviously many awards to his name. Subsequently joined the Faculty of Physiology in the same university, where he became Professor of Physiology. He had a 3-year stint in UK, where he did his doctoral thesis on growth hormone. And then when he came back, he started a study group on hormones and its action. And the physiology department then also had others who were interested in hormones, leptins, growth hormones, etc. And Professor Carlo's team founded the Endocrine Society of Sri Lanka in 1979. And as has been said earlier, he was the founder president of the society. So while in UK, Professor Carlo also discovered that in order to treat people with growth hormone, you needed to extract pituitary growth hormone from their brains, a practice that went on till the CJD epidemic in UK. Until then, they would do the same in Sri Lanka, extract the pituitaries from cadavers, export it to Denmark, and then get in return funds as well as growth hormone back. And they would run a growth hormone clinic in the department. So we are here today, grown from those founding days into a proud college, and I'm sure he would be proud. So in his memory, I'm here to talk about viruses and hormones, and I'm sure he would be inquisitive about what I'm going to speak, and he would have been surprised to see that IGF, further hormone down the line of growth hormone, could actually be produced by viruses, and they can be made to manufacture IGF. So there are many questions about viruses and many sentiments expressed about viruses, and I'm sure most of the audience would say we would better have a life without viruses. A world without viruses would be a better world, you would say. Can I ask you to do an exercise for me? Take a deep breath in and then let it out. Now, in that deep breath that you took, 50% of the oxygen came from the marine environment, and those viruses were responsible for that 50% of the oxygen you just breathed in. And that is because viruses in the ocean make phytoplanktons and cyanobacter produce oxygen by taking carbon dioxide and do photosynthesis and create the oxygen back into the system, in that process also recycling half of the world's carbon dioxide. What the viruses do is insert genes into those unicellular organisms and make them do photosynthesis faster and better. If it is not arrested, those very organisms will use that energy up and release carbon dioxide back. But the viruses block that process. So you have carbon dioxide dumping into the ocean, and that carbon material is then recycled up to the whale. So obviously the food chain starts from the phytoplanktons and the cyanobacter. And so whatever you see in the ocean starts from there. And whatever the air you breathe, half of that oxygen, the viruses are responsible for. Actually, when you last waded in the ocean, you waded through millions of viruses, as high as 100 million per teaspoon of seawater. And if you put all of the viruses in the ocean together, they say it would weigh more than 30 million blue whales. Nobody has had a scale to weigh that, but that's the estimate. If you put all the ocean's viruses together, you could build a bridge up to the next galaxy. And that is how many we have. So if your school taught you to read this number, 10 to the power of 31, that's many viruses we have in the world. And that's read as 10 quintillions, or to understand it better, 1 million trillions. So 10 to the power 31. So the ocean and the world are full of viruses. And we seem to live in an environment that is enriched and is owned by viruses. And that's the complex things that viruses do in the oceans. My next question to you is, did you realize how much of you is viral? I know that behind your masks, you think you are in a virus-free environment, but did you think about the human microbiome? So if you take the feces and study, we know that there are 100 billion virus particles per gram of feces. Just to give you a comparison, there are 30 trillion cells in your body. And there are, in the human microbiome, 38 trillion bacteria in our gut and in other epithelial surfaces. If you count the number of viruses, you find that viruses are everywhere. They are also in the gut, but there is not a single place that doesn't have a virus. And if you look at the blood, if you look at the brain tissue, nervous tissue, and the junctions, the bones, viruses seem to be there, and we actually don't know what they do. So many of you I know are proud recipients of microbiology distinctions, but now you have to go back and learn your microbiology, because when they taught you, we didn't know about viruses and what they did to us. So if you look at the human gut virome, as I said, the viruses are everywhere in the body, but if you just look at the gut virome, there seems to be a very fixed pattern for an individual. In childhood, there seems to be one set of viruses which transfer into another set at adolescence, and then a more mature adult would have a different set of virus particles. What they do and how they contribute to illness or health, we do not know. They seem to be in symbiotic relationship. We also know that they can transfer particles and proteins into other bacteria and other organisms. So we guess that they must be inducing certain symbiotic functions in bacteria. This is one of the visions of how this could happen, that the viruses and other nematodes and the bacteria in the gut, on the top left corner, Joanna, by the way, drew this beautiful cartoon, can induce immune response through the T cells and can actually contribute, they say, to innate immunity. And so what we are today, our defenses were partly coded for by the viruses in the gut, along with other organisms. They also may be inducing inflammation and other illnesses, and we have very little knowledge about that. As I said earlier, viruses are prolific, they are everywhere. they are also carriers of genes and are adept at drilling into cells and horizontally transfer genetic material. So vertical is mother to fetus, horizontal is that suddenly an organism could be gifted with a new set of DNA or RNA that could code a new function. As I said about the cyanobacter and phytoplankton, and so this is what we learn in microbiology, that there are multiple mechanisms through which viruses or bacteriophages, the viruses that infect the bacteria, can transfer material. And we know that for multiple reasons, for example, the drug resistance in bacteria could be induced by the virus. And so viruses are the builder of the web of life. They can suddenly transfer a genetic material where it's least expected. So they can promote adaptation. As you can see here, when they infect and give a horizontal transfer of genetic material into a cell, you have new function and new protein synthesis that happens in that cell, whether it's a monocellular or a multicellular organism, you have that process. So you start producing new genes, you can have a new adapted molecule, and the new advantage in a suddenly dormant organism that you can lead to selection or deleterious effects in that organism that leads it to die. But viruses usually would want their host to be alive. Humans seems to be very happy with the viruses. In fact, all viruses are happy with the humans. This is a picture of the 21 chromosomes and the many receptors that our genes code for, which the viruses bind to. For example, the herpes virus has multiple receptors in all over the chromosome where receptors are produced. And our friend, the coronaviruses have a chromosome 15 based mechanism where they code for a particular receptor and so on. I showed you how our cells are 38 trillion and the viral load in our human being is much more than that. If you look at the genome of the human, we know that only 2% in that light orange are protein coding genes, and that is all we have. And that's all the functions that we know come from those 2%. The rest of the 98%, if you look, 10% of them are actually viral genes that are in our genome and are vertically transferred from mother to fetus and the fetus to the next fetus and so on. So we have 10% of our genome, which is actually composed of viral material. Most of them is retroviral. For obvious reasons, retroviruses put their gene right back into human cells. And then there are 2% of other viral. There is a dark matter of 88% where we don't know what the function is. There are non-coding genome, but in future, you will find that lots of these also are partly viral. So they reflect past infections and past transfer of genes into our cells. If you don't believe me, you can look at this example. So what would be the difference between an egg-laying dinosaur and a placental human being who is born through placental conception and conceiving? Well, in a reptile or a bird, the fertility happens by laying of an egg outside of the mother's body, and then it has to hatch. Whereas in a placental mammal, the fetus becomes a parasite of the endometrial tissue. It has to now start invading, form trophoblasts, and start sucking out nutrition from the mother. How would you transfer from being, and what is the difference between an egg laying and a placental human being? In order to do the function of invasion, the specialists are viruses. You need to invade the host without the host knowing that it's happening. And that is done by the syncytin-1 receptor. And that is what forms the syncytiotrophoblasts, which are the invaders into the endometrium. Guess where it came from? The specialists of that function are viruses. And so if you study the syncytin, you will see that it's actually a herpes virus coded gene that had been inserted into our ancestors millions of years ago. So if you are alive today, and that reproduction process, viruses have been responsible for. If you look at the way we live alive, so normally a normal cell would remain strong and without any mutations within it. Should the mutation occur in the middle graph, a p53 protein gets activated and the cell's division is arrested. So if you have a mutation, that cell shouldn't divide, and the p53 is responsible for that. Whereas if the repair doesn't occur, that cell will lyse. If the repair had occurred, p53 will degenerate and the cell will then be allowed to regenerate. So that p53, the master regulator, which is actually an anti-cancer mechanism within our gene, is actually a viral protein inserted into an arthropod. I can keep talking about how viral genes are within us and how we are made up of viruses, but I'm going to move on to my next topic. So hormones made by viruses is an interesting emerging topic. Before I go to viruses making hormones, I want to show you a video of how insulin, a hormone, is used as a weapon.
Snail goes spearfishing. Its extendable proboscis is packing a concealed weapon, a tiny hollow harpoon made of chitin, the same tough stuff in a lobster shell. And the end of the proboscis is tricked out with receptors, taste buds, that help it close in on its target. When it strikes, the snail's pace jumps to light speed. The embedded harpoon doubles as a hypodermic needle to inject the victim with paralyzing venom. As it reels in the catch, the cone snail uses...
All right, this cone snail uses a modern version of insulin. So if you look at the cone snail structure, you can see that at the bottom you see the human insulin structure. Just like the human insulin, it has an alpha and a B, A and a B, and only thing that it misses is this long B chain. Instead of a 30 amino acid, it has a 21 without that hook, which is responsible for forming hexamers. So if you have such an analog or a modern insulin, as you can imagine, it is rapidly acting, and you saw the effects. And you can also, by the way, see the impact of where the needle is not seen, that kind of an injection device that it has. It's so advanced that the fish die almost immediately. So insulin can be used as a weapon. And thinking on those lines, these researchers, Altindis and others from Harvard and from Japan and many other centers, asked this question: are there viruses that are producing human-like hormones in the environment, and could that be impacting our health? So what they did was to look at, as I said, there are millions of viruses, but we only have the full genome sequencing of a very tiny proportion of 7,500 or thereabouts. So they asked the question, do they code for 62 human hormones and adipokines and other intracellular paracrine secretions? And they found that a large number of human hormone-like particles or peptides were coded for among this lot of 7,500 viruses. What they wanted to see was to ask, are they actually very specific? And if you code the 3D structure, will it work just like the human hormone? So they came up with two shortlisted individuals, iridovirus, which causes fish infection, and lymphocytic disease virus, which also causes fish disease, that secreted just like the human insulin and IGF looking structures. And you can see how closely 3D wise it resembles. They didn't stop there. They did functional studies to prove that these two hormones, the VILPs, virally induced insulin-like peptides, they call them VILPs, just had the function of human insulin and human IGF. So the hypothesis is that the infected cells by these viruses could give rise to endocrine and paracrine signaling and do what IGF and insulin can do. They can cause hypoglycemia that we don't know about. They can cause insulin resistance. They can cause tumor formation due to paracrine action of that IGF over in that area. So I'm going to quickly move to my next question. There is a saying in the nursery rhymes we were taught that what are little boys made of? They are made of snails and slugs and puppy tail pieces. And what are little girls made of? Sugar and spice and all things nice. And if you come to influenza, it seems to be correct that men get the worst of the influenza and they get more severe illnesses. Even in COVID, we know that the ratio of mortality is 3:1 for men compared to women. And if you look at influenza severity in pandemic as well as seasonal, the solid ones are the men. You can see that for both types of epidemic or pandemic, the men get it worst. Why would that happen? Well, if you look deeper, you can see that on pink on top, that's a busy cartoon, but just to highlight few facts. Interferon, you know, is now known as anti-influenza, anti-COVID effective medication. And interferon is increased by estrogen. Also, the defensive interleukins are increased by the estrogen, whereas men seem to be creating the hyperinflammatory hyperimmune syndrome by producing more tumor necrosis factor, more interleukin 6 and 10, which are all markers of severe disease in COVID and influenza. So estrogen seems to have a protective effect against viral infections, at least respiratory infections. So moving on in my last 5 minutes, I wanted to highlight the pandemic, and who can stop talking about viruses without talking about a pandemic. The last decade, this is not this year's document, last decade, there had been lots of documents that have highlighted the risk of a pandemic almost imminent. And they highlighted that these 4 characteristics are required for a virus to create a pandemic. It has to be respiratory because it can rapidly spread. It has to have silent carriers, or it can spread in incubation period. And it has to have no host exposure or no previous exposure to that virus and highly virulent. So those 4 characteristics are required in an organism or a virus to cause a pandemic. So we had 2 SARS-CoV epidemics earlier. the SARS outbreak in the early 2000s and the MERS-CoV outbreak 10 years or 8 years ago, we have this time have a virus that is a fairly advanced version of the previous ones. It has more specific receptor binding to the ACE2 receptor, which is found all over the body. Once again, it's not just a nasal or a respiratory receptor, it is found all over the body. So it binds to the ACE receptor TIM-PRES complex, and you can see the advanced technique that it employs to enter the cell by first modifying its own S1 by binding to the TIM-PRES. That enhances the ACE2 binding, and then the virus is taken in. So it's not just a tiny unknown pathogen, it's very highly specific killer machine, because it has a very specific binding, and it has found a receptor that is all over the body. And ACE2 is found all over the body, and it can insert its protein into almost every tissue to produce copies of itself. So you have seen this image of how it inactivates the ACE2 by that binding system, which in return leads to higher levels of the pathogenic angiotensin 2 and doesn't allow the deactivation of the angiotensin 2. That leads to hyperinflation, inflammation, and tissue damage. Incidentally, 2020 is the 20th anniversary of the discovery of the ACE2 receptor. It was discovered in the millennium. So in our group, we asked this question: if ACE2 is such a vastly spread out receptor, could it be involving endocrine organs also? So far, not many studies have been published on that. So all these organs express the ACE2 receptor. And we started going through the literature over the last 2 epidemics emerging of similar virus to ask whether that had caused organ dysfunction similarly. So we figured out that almost all of the organs could be affected by the viruses, and that resulted in our review that has been published last week, called The Impact of SARS-CoV-2, and you can see the media interest that it has created. There are more than 100 media reports of that internationally in multiple languages, and currently we are the featured article of the journal. So what we highlighted in our work is that COVID can affect all of the endocrine organs. It can cause hypothalamus-pituitary damage, causing hypothalamitis or hypophysitis. Actually, it has been known that lots of patients and survivors have a post-viral fatigue syndrome. Lots of them could be due to the ACTH deficiency that results from that. We also know that the COVID mimics ACTH molecule at least in 34 areas of its structure. So it might be mimicking ACTH and causing autoimmune destruction also. We also know that the previous epidemics cause adrenalitis, and therefore treatment of patients is important. We also highlighted that koala syndrome. Koalas don't have cortisol response because their adrenals are almost non-existent. So in those who have critical illness related corticosteroid insufficiency, we need to recognize that and treat. And this is what we highlighted in that article. And as you have seen, dexamethasone has been shown in at least one study so far to be an effective therapy. And long before the trial was published, we had predicted that that would be beneficial. We also have 1 or 2 case studies just emerging that COVID-related thyroiditis is true. And we predicted that all of these mechanisms, destruction, subacute and acute thyroid syndrome, could all be seen in COVID patients. It can also cause pancreatitis and cause sudden onset diabetes by destruction of the beta cells. And we also finally showed that vitamin D deficiency seems to be an important marker of the severity of the illness. And because, as you can see in the ACE1, ACE2 system, vitamin D is an integral part of that mechanism. So, ladies and gentlemen, my time is up, but I have come to my last slide, and I have 6 take-home messages in the memory of Professor Carla Fonseca today. So my question now is, are they friends or foe? I'll ask you to think through that. But I wanted to remind you that the viruses are ubiquitous, they are everywhere, they are not just pathogens, they are ecosystem stabilizers. We are alive today, perhaps because there are viruses active in the ocean doing all what they do. We also saw how Virome in the human body, viruses are key players, and we know very little about all the viruses that we have in our system. And lots more work has to be done about understanding autoimmune endocrine disorders and other disorders. I showed you examples of how horizontal gene transfer can happen and how we are alive today because of that process of how viruses cause rapid advances in body functions. Suddenly, we looked at estrogen and their protective role against influenza at least, and estrogen as a defender and testosterone being poor in that aspect. I showed you how hormones can be made by viruses, and we know very little about that field because out of the millions of viruses, only 7,500 were properly studied. So we don't know how many more are there, and whether all the hormones we have come from the peptide hormones, have come from viruses, is left to be seen. And my last message is our publication that our group has worked on, is to remind you that SARS-CoV-2 can cause endocrine devastation. It can affect almost every organ that produces endocrine hormones. So thank you very much. And I thank the SLCE and the president and the council for having invited me to deliver this lecture. Thank you.